The third speaker in this remarkable segment is somebody who has been studying NNR, neuro-nano robots. This is not science fiction. This is something he has just completed a remarkable PhD in North Portugal. He is en route to continue this remarkable work in Berkeley. This is Dr. Nuno Martins. He is not just an academic by no means. He is also a serial businessman and entrepreneur. He has created a number of companies based on some of his ideas. He is also very much a person who practices what he preaches. He has been concerned about longevity all his life. He has become a trainee tennis coach, a trainee swimming coach, a fully qualified swimming coach, a fully qualified tennis coach, a fully qualified power lifter, dietary expert. He is really a Renaissance man, a polymath. Please welcome Dr. Nuno Martins.
We're going to talk about neuro-nanorobotics. This is a technology I've been working at for 10 years now. It's a proposed medical technology to preserve human brain information. What's the goal? The goal is to treat brain disorders. When we look at this challenge, we realize there was a necessity for more data. Current strategies to extract human brain information in the clinical practice are, for example, MRI, magnetic resonance imaging, fMRI, functional magnetic resonance imaging, for structural data and functional data, electroencephalograms, and PET scans. These are 4 examples. At the research level, some researchers are using microelectrode arrays. It has been used to extract data from a monkey's brain, extract the data to a supercomputer, and control a robotic arm. None of these technologies can be scalable down to a level that allows us to preserve the fundamental human brain information comprehensively for the whole human brain. So we're proposing a new technology called neuro-nanorobotics. This is an electron microscopy, and highlighted in blue is a neuron. And the arrow points to a neuro-nanorobot that will monitor the action potential of a neuron. Not just the action potential, but its waveform. This is a neuro-nanorobot on a glial cell. So the brain has 2 main types of cells: glial cells and neurons. Glial cells do not process electrical information, but they're involved in several brain disorders, so we need that data. This is another type of neuro-nanorobot monitoring synapses. Neurons and synapses are believed to be the processing units of information on the human brain. 90% of a human brain synapses occur in spines. These are the 4 most common spines. And in these 4 electron microscopies, you see the neuro-nanorobots monitoring the action potentials being processed on the synapses. In 2012, we published a paper called Non-Destructive Whole Brain Monitoring using Nanorobots, Neural Electrical Data Rate Requirements. If we want our neuro-nanorobots to extract the brain data from the patient, we need to know how much data we're handling. So we looked at the literature, and the estimates were very different. The minimum was 1.48 times 10 to the 11 bits per second. The maximum was 32 times 10 to the 28. So the best estimates had a difference of 17 orders of magnitude. So we needed a more precise estimate. So in our paper, we developed this formula, and we used it to estimate the quantity of information being processed on a whole human brain in all its subregions and all its regions. This is part of that table. In this table, we have the number of neurons for each region and subregion of the human brain, the number of synapses, again, for all the regions and subregions of the human brain, and also the percentage of the different types of firing patterns on neurons. We have regular spiking, fast spiking, and bursting. These are the three main categories. On the last part, which you probably can't see there, the last column has the quantity of bits per second being processed in each subregion of the human brain. So for the brain as a whole, since 2012, we have a precise estimate with margins of error of how much data we're handling. So the neural nanorobots will be extracting and transmitting into the supercomputer 5.52 plus or less 1.13 times 10 to the 16 bits per second. This is the processing capability we need. So finally, we knew how much data we would be handling. But to detect that data, we need nanosensors on board of our neural nanorobots. So in another of our papers, we looked at the different possibilities. The first type of nanosensors we look at were concentration-based nanosensors. They count molecules. And we realized they could not provide the necessary spatial and temporal resolution that we require. So we looked at a different type of nanosensor called passive fat-based nanosensors. This seemed to be the right solution. They could provide the proper temporal and spatial resolution, but there were size constraints. They needed to be of a certain size, and we could not accommodate enough of these sensors on board of our neural nanorobots. Finally, we discovered the strategy. They're called active fat-based nanosensors. So we designed the nanosensors, and we published them. So these nanosensors can be used, and we can accommodate enough of these on board in order to detect each single action potential and its waveform for every single synapse and every single neuron on a human brain. This is an example of three of those nanosensors on board of a nanorobot. They're inside the neuron, monitoring the action potential next to the neural membrane. Even if all the neural nanorobots and all the nanosensors get damaged, the single-wall carbon nanotubes released into the cytosol would not compromise cell viability. In summary, this is a safe technology. 20 to 30 years from now, when a patient gets in the physician's room, he will simply sit down, receive an injection, and the neural nanorobots will navigate through the vascular system, cross the blood-brain barrier, enter the brain parenchyma, navigate intracellularly into the neuron, glial cell, and synapse, and report that data into the supercomputer on the physician's room. That's good. A few minutes later, the neural nanorobots are removed according to a certain protocol, and the patient can go home, and all the data that it's necessary to properly evaluate and use the proper strategy to cure the patient is available to the physician. 15 minutes are not enough to talk about all the subcomponents of the neural nanorobots. We did talk about sensors. We did talk about brain data, how much brain data we would be reporting. We would have to talk about locomotion systems for each near nanorobot, onboard computing systems, power systems, data transmission, and data storage and processing on the supercomputer that stands outside in the physician's room. Once in the clinical practice, this technology will be working in parallel with supercomputing, artificial intelligence to handle and process the data, and atomically precise manufacturing, among other technologies. All of these technologies are expected to grow exponentially, 20 to 30 years. We believe this technology may allow us to treat the approximately 400 brain disorders, such as Parkinson, Alzheimer, depression, addiction, and others. Thank you. And we also believe this technology may have several non-medical applications, helping us solve some of the most difficult challenges we face today in education, research, communication, and other current problems. This event accommodates amazing people from 30 different countries. So to finish, I wanted to be sure that I thank you, each one of you, in your language. So thank you. That was good. Thank you.